My name is Catherine, and I'd like to call up Natalie McKenna to do the introduction for today.
Thank you, Catherine. Thank you, everyone, for being here. For those who don't know, the Hariri Institute's Junior Faculty Fellow program really aims to recognize the outstanding members of the VU community who are in their early career in computational and data research at VU. And we also aim to connect them with one another in the Hariri Institute community at large. So thank you for everyone for coming and connecting with Josh this afternoon. I also wanted to just point out that as part of their fellowship, each JFF does give this signature 45 to 60-minute talk to our diverse constituency here at the Institute. So with that being said, I will hand it over to Joshua Campbell. Thank you.
Great. Thanks, everybody. It's a pleasure to be here and present in front of you guys. I recognize a few faces, but I'm excited to meet a few more. Right, so my background is in computational biology and bioinformatics. I actually did my PhD just down the road in the VU bioinformatics program, so I spent a lot of time over at the ELSA building. I did a postdoc at the Broad Institute, where I worked on cancer genomics, and then came back to faculty in 2016 in the section of computational biomedicine in the Department of Medicine. Since then, I've been working on developing methods for this new technology, which is really popular in the field of biology, called single-cell RNA sequencing. Okay, so just out of curiosity, has anybody heard of this before? Frederick, you're not allowed to raise your hands because I'm working directly with you. All right, so it's no worries. It's been very popular. So you guys know about the journal Cell Science and Nature. So this is sort of a quick inventory that I did for 2018. And if you look at the number of publications that use single-cell RNA sequencing to discover something new in biology, the cumulative total was about 65 publications across the three journals. And in Science's, their breakthrough of the year, they highlighted this paper where they did single-cell RNA sequencing of zebrafish embryos to understand more about development. So it's been very impactful and very important in the field. Question is really why? And so I wanted to give you guys a little bit of a background about sort of the perspective that we're coming from and why we think the technology is working so well. And then I'll get into the algorithms and the computational work that we're doing. So it's interesting, the answer to why single-cell genomics are important is actually already answered a long time ago on Wikipedia. So this is if you go to this particular page, it talks about biological organization. And there's sort of a fundamental concept that we use in biology to help us sort of conceptualize things, and that's that biology can be organized into different hierarchies. And each level within that hierarchy is sort of composed of different subunits that sort of work together to cooperate, or perform particular functions, or do particular behaviors. So if you kind of start at the top of the hierarchy, you have the atoms, oxygen, hydrogen, whatever, and they bond together to form different molecules, and those molecules perform different functions. Those molecules then form macromolecules. So this is something like a DNA or a protein molecule, and those again perform more macroscopic functions. Those guys organize themselves into cells. Cells can organize themselves into tissues, and tissues into organs, and this whole thing actually goes on like ecosystems and a little bit beyond the scope of what I'm doing today. Although, yeah, if we went up to single cell of ecosystems, we definitely need Spark for that. So maybe that could be a future collaboration. But within each one of these levels of the hierarchy, it's important to have sort of a catalog of the different subunits that are there and sort of what their function or behavior is. So you can think of something like the top level there, the atoms. We have the periodic table, very important for chemistry. There's a certain number of elements that can be studied. You can go sort of the other flip side of things, and you think of anatomy and physiology, understanding what all the tissues are, or the organs are in the human body is important for understanding how the whole human body works as a whole. So this is a initial drawing done by Leonardo da Vinci a long time ago. So each sort of level has its own kind of expertise and set of subunits or components that is trying to be characterized. So we can, we're talking about single cell RNA sequencing today, so we can ask ourselves, well, what's going on at the cell level? Biologists have been characterizing and discovering cells for a really long time now. So this is, Robert Hooke was one of the guys who discovered the first cells, and he looked underneath the microscope at a cork, and you can see that they were organized in these little squares. That's where the term cell comes from. He named them. But since then, we've used things like microscopy to look at cells in all kinds of different organs and tissues, and we try to characterize them in various ways: their shape, their size, maybe what we think they're doing in the tissue. But what would actually be even more powerful is a potentially way to do it in sort of an unbiased fashion, where we can look at what a cell is composed of, maybe look at all of its subunits, and then look at the combination of those that define each particular cell, and that might give us sort of a broader classification, a better periodic table of cells in a way. So to do that, I'll tell you how single cell does that in a minute. But first, I'm figuring a lot of people here probably aren't biologists, and so I'm going to just give you a real quick reminder of what you learned probably in your freshman biology class and quickly forgot. And it didn't matter anyway because everything in biology is now in Wikipedia, so it's fine. So if you remember this concept, the central dogma of biology, where you have your DNA. Of course, everybody probably knows what DNA is. We hear it all the time on CSI and the other shows. DNA is composed of these 4 bases, which we code them as A, C, T, and G. Your DNA sequence is about 3 billion bases long. But there are parts of the DNA which are called genes, and these genes get transcribed, they get copied. So if you think of DNA as the code book, this RNA is a copy of one of the pages in those books. The RNA then codes for something called a protein, which is made up of amino acids. And these proteins are just like little molecular machines. They go and they do stuff in your body. They build stuff, tear stuff down, hold stuff together, everything. So that's kind of how the code works. And so we can, especially, what's interesting is that every cell in your body basically has the same underlying genome, so it has the same underlying DNA sequence. But clearly, not every cell in your body is the same, right? A skin cell is different than a lung cell, different than the cells in your eyes. So how do cells be different from one another? Well, they sort of turn on different sets of genes. So there are about 20 or so thousand genes that people have characterized in your genome. Here I'm just showing you a simple cartoon with 3. But each gene can be sort of transcribed in a different amount. And so in this particular example, we have 5 copies of the red gene, 2 copies of the green gene, 1 copy of the orange gene. And let's say these genes are involved, something important for nerve cells. And so this combination of genes will then define or make up a nerve cell. But a different combination of genes, let's say this is something important for immunity. So this combination will make up a lymphocyte or an immune cell that you see in your blood. So it's really these sort of combinations of genes that allow us to define these unique cells and cell types. And what's interesting, there are methods now where we can sequence a given sample and detect all the genes that are present or turned on in that sample. We've actually been able to do this for 10 or so years now. But the problem is we haven't been able to do it at a single cell level. So what we have been able to do is take a tissue sample, whether it's cancer or some sort of organ in development or whatever it is. We grind up all the cells, you get this pool of RNA, and then we can sequence it and determine, there were 6 green transcripts in this sample and 2 red ones, and or 2 genes detected in that sample. We can do another sample and we can say, there's fewer green genes detected in this sample, but we can't really know what cell those genes were coming from. So if you think of this as a data matrix that comes out of the end, each column here is a sample, like a whole tissue, and then each row is a gene, and we're counting the number of genes detected, or number of transcripts detected in each sample. What single cell allows us to do, and I'm totally glossing over the technical details. There'll be a little bit at the end when I talk about sort of future technologies, but essentially, conceptually, what it allows us to do is do the sequencing of each individual cell now. So now we can determine, in this particular cell, we had 3 red transcripts, 2 yellow transcripts, and in this other one, we had 3 blue transcripts. All of our green genes were actually coming from this cell type, which maybe we didn't know about before. And so our sort of final data matrix looks something like this, where for each sample that we sequence, we now have multiple columns. Each column corresponds to a single cell. And then, again, we have our 20 or so thousand genes, and it's the count of each gene in each cell. Does that make sense to people on a sort of conceptual level? So if we sort of go back to our hierarchy here, the problem is when we were doing bulk sequencing, we were sort of missing this level in the hierarchy. We were detecting the level of RNAs and sort of trying to make inference about a given tissue or a given sample, but we were really losing the cell information. And really, single cell allows us to capture what each individual cell is and be able to begin to understand what it's doing. And then we can look at sort of differences in abundance of different types of cells across our different samples. So now moving a little bit into the computational problem and the algorithm that we developed. I'll give you a quick overview of that, and then I'll show you an actual data set we generated and analyzed. So what's interesting about sort of the data that comes out of it, I showed you kind of the raw data matrix, what it looks like when we get out of it. There's some features about it. One is it's very discrete, so we have integer counts of each individual gene and each individual cell. And it's pretty sparse. So if you think about, in reality, a cell may have several 100,000 transcripts in it. But due to the limitations of the technology, we're only taking a very small sample of that. So maybe we're getting 5,000 transcripts to text. So, column sum for a given cell might be, 5 to 10,000, something like that. So we're just getting kind of a very small snapshot of what's actually in that cell. And so the data tends to be sparse, have lots of zeros in it. Another feature is that cells with similar patterns, we assume that there's a lot of redundancy, so it's not that every single cell is going to be completely unique, but all of your cells of one type, all of your skin cells or whatever, are going to have a very similar expression pattern. So we can actually do clustering. So even if each individual cell has relatively weak signal, we can kind of cluster together cells that have similar signals and get a better picture of what that cell population is. So that's a huge part of what single-cell sequencing analysis is sort of clustering cells based on their gene expression pattern. The other thing that's interesting, which is we sort of try to highlight in our algorithm more than others, is that groups of genes will sort of covary across cells. So it's not that every single gene is off doing its own unique thing, but there are groups of genes that are involved in the same biological process, and they sort of get turned on or off together in different groups of cells. So we can actually do clustering on the genes or clustering on the rows of our matrix as well. Say, hey, these groups of genes have very similar patterns, and that means something biologically. So when we were developing new algorithms or thinking about ways to approach this data, we actually took inspiration from the field of topic mining or natural language processing, NLP. Is anybody here familiar with NLP or have done stuff with NLP in the past? Okay, a couple of people. You guys ever heard of latent Dirichlet allocation? A little bit? All right. So it's an interesting algorithm. There's some similarities to what we wanted to do, although we end up tweaking. I'll show you our tweaks in the next slide. But conceptually, it's you treat each document as sort of a distribution of words or a bag of words in the same way we kind of treat ourselves as like a bag of genes or bag of transcripts. And each document has sort of a different distribution of topics or themes. And so in this particular document, there's a high proportion of this yellow topic, and each topic itself is a combination of different words. So the yellow topic is talking about genes and DNAs and genetic material. The blue topic is talking about computational stuff, data, number, computer. And then there's, in this particular document, there's no green, so you don't find any words related to brains. It's not a neurobiology document. If you were to go to a neuro document, maybe you would see higher levels of this and this and less of the yellow or something like that. So each document can have its own sort of different distribution of topics. We sort of leverage this idea. The algorithm works by essentially looking at co-occurring word counts across documents to identify these topics. We leverage that idea to find look for co-occurring gene counts across cells to identify these sort of modules. And then we also add an extra layer of clustering on the cells themselves, or we cluster documents together that are similar to one another. So what does this actually look like? Again, if I'm sort of recapitulating this hierarchy in my own way, we have samples or tissues that each themselves are composed of different proportions of different cells. And each cell itself, so in this case we have red, purple, brown cells, and each cell itself is composed of different groups of genes. So we have this blue group of genes, which all tend to covary across the different cell populations. So they're turned on in the red cell, turned off in the other two cell types. And each cell type is a mixture of these different groups of genes. And then each group of genes itself is a mixture of different genes. So blue gene is, has high levels of A and B and sort of medium levels of C and D. And you can see those same relationships all the way through. And when we get our data, we just sort of have the raw counts, and we're kind of working our way back up the hierarchy. We're trying to group genes into these modules, determine what combination of modules make up a particular cell group, cell population, and then what are our frequencies of our cell populations across different samples. And so we have this discrete Bayesian hierarchical model. I'm not going to go through the details of it now, but if anybody's curious, I'm happy to do it afterwards. Essentially, it does two things. It serves as a bi-clustering method, so we're both clustering the columns of the matrix and the rows at the same time. So Y is our sort of module that each gene belongs to, and Z is our population or group cluster that each cell belongs to. The other thing that it does is sort of gives us this matrix factorization, so it kind of factorizes the original counts matrix into these three probability matrices, and they're sort of useful for exploring the data downstream. So we get the probability that each gene is a part of each module in the bottom matrix. In the middle matrix, we get the probability that each module is turned on in each cell population, and then in the top matrix, we get the probability that each cell population is present in each tissue or sample. This middle matrix, I'll show you examples of it later, how it can be really useful to give you sort of a reduced representation view of your data set. So you just kind of want to know what sort of biological processes are present in each cell population without having to look at 10,000 cells or 20,000 genes at a time. Maybe just a quick toy example to give you a better sense of what the algorithm is actually doing. So if we have a very simple counts matrix here, so we have different samples, 1, 2, and 3. Each one has its own count matrix. Each row here is a gene. Each column is a cell. And so you can see here that these first three columns here have very similar expression patterns, or they have very similar count patterns, so they get clustered into a group. Next three cells are very similar patterns, so they get clustered into a group. The last three cells get clustered into a group. The genes themselves, you can look across this way, and you can see that these genes, even though this gene is expressed higher than this gene, they get turned on or off in the same amount across the different clusters, and so they get grouped together in a module. These next three genes are very similar, so they get grouped in the module, and then there's these last two groups of genes that get clustered into modules. And then the very top is sort of the useful matrix for kind of this. It's a reduced view of this big matrix where you can see essentially, for that first cell population, the orange one, this module is turned on really high, this one's turned on at a medium level. You can just see it by looking at it without having to go through all the genes themselves or look at every cell. And you can see basically each cell population is itself a different combination of these modules. So the benefit of this model is, as I mentioned, can do the simultaneous wide clustering of the genes in the modules and cells and subpopulations. We talked about the sparsity a little bit. A lot of the other algorithms that analyze this data have to do like a lot of filtering or massaging of the data to handle all the zeros. Our model doesn't necessarily mind them, so we can keep more genes in our clustering. We don't have to exclude them, and it allows us to get a sort of more fuller picture or better picture of what the cell looks like. And then something that's useful, we don't have to do a lot of normalization on the matrix. We can just take a raw count matrix and sort of feed it right in. All the other packages out there that do like normalization and error correction and all these other things before they actually get to the clustering, which can introduce biases. All right, any questions about the model or anything like that before we move on? I'll show you an example. Yeah, sure. Mm-hmm. Yeah, it's a good question. Each gene, so homoscedastic nature, it's the sort of absolute level of each gene might be different. So you might have a gene that has a lot of counts, a gene that has low counts, but they sort of go up or down by the same amount across different populations. So that tends to be a little bit more homoscedastic in a way. So I'm going to show you an example where we were looking at the effects of cigarette smoke on human airway epithelial cells. So everybody here has an airway, turns out. So this is, you think of your airway as a tube. It's lined by a particular set of cells. It's called an epithelial layer. It helps sort of protect your airway against toxins and pollutants and pathogens and other things. This lining, this epithelial lining, is called, or is composed of different cell types. And there are many that we already know about. So you have, for example, different kinds of secretory cells. So here we have two different kinds shown, called a club cell and a goblet cell, but they each secrete a different kind of mucus, and the mucus is used to capture particles that you may be inhaling. You have your ciliary cells, which are in orange here, so they have little hairs on them that sort of all beat in synchrony, and they help sort of move particles up and out of your airway. So they're all of these cells together help perform mucociliary clearance. That's what gives you your coughs and other stuff to help get that stuff cleared out. There's also a type of cell called a basal cell. So these cells can either divide into new basal cells, or they can, it's called differentiate, and basically turn into the other cell type. So a basal cell can become a ciliary cell or become a goblet cell. So that's how your airway replenishes itself over time. So these cells are replicating and then replacing the other cells that die off. There's actually more cell types here, but I'm not going to, in the airway, I'm not going to go into it for the sake of time. But we worked on this project with Avi Spira from our section and Jennifer Bean. This is kind of a follow-up to one of the studies that they did several years ago now, back in 2004 and 2007. They were actually really interested in what genes get turned on or off in response to cigarette smoke exposure in the airway. And so again, here they were doing bulk. They weren't doing sequencing, but they were doing something else, another technology called a microarray, but it's essentially like bulk sequencing, where they were taking a brushing of somebody's airway, measuring the genes that are turned on or off, like across the whole, all the cells in there, and trying to figure out sort of the biology. And that's what this heat map is showing you. So here, each column is a particular patient or brushing from a patient. Each row is a gene, and if it's more red, that means that gene was more turned on in that sample compared to the other ones. And so all the samples from current smokers are over on the right, so you can see the set of genes were all turned on in the smokers, and lower or turned off in the former and never smokers. Whereas the genes down here at the bottom, they were all turned off in our smokers, about higher levels in the nevers. And, you know, these genes made sense biologically. There were a lot of detoxification enzymes, so things that you'd expect to be turned on to sort of handle carcinogens and stuff like that you get exposed to when you're smoking. So for a long time, that was kind of the story. Maybe 5-10 years, it's like, okay, you turn on detoxification genes, boom, great, we know everything there is to know. But it turns out if we sort of, even at this bulk level, if we take a little bit of a deeper dive, there's some indication that that's not the entire story. So this gene called SCGB1A1 is a marker. It's expressed mostly exclusively in club cells. So it represents one of our particular cell populations. And it's going down. The other gene up there, MUC5AC, is the marker. It's expressed in goblet cells. So this other kind of secretory cell. And so it could be that all cells within that brushing are just sort of turning on these genes together and turning them off altogether. Or it could be that we're actually seeing a higher proportion of these different cells appear in our sample. And we couldn't really tell that with this particular data, but it gave us the indication that we maybe need to take a deeper dive, that there are more processes going on here than what we really understand. So that's when we decided to do single cell. So we use this to get brushing of your bronchus, your airway. There's a bronchoscopy procedure where they have a tube with a camera on the end of it, and they'll stick it down past your trachea into your airway and take a brushing of the right mainstem bronchus. We have studies ongoing where we need volunteers, so if anybody wants to do this, you're totally welcome to. I've actually done it before. It's not that bad. Sounds like people don't believe me. Actually, okay, so the worst part, you don't have nerve endings in your airway, so you actually can't feel what's going on. But the worst part is they have to stick the tube past your gag reflex and so back to your throat. So they have to spray lidocaine in your mouth, which just tastes terrible. So I was gagging and crying on the lidocaine. But then after 10 minutes, you can't feel your tongue, you can't feel the back of your throat, and so then it's just whoop right down and in, and it was fine. But it's also weird because then you can't feel yourself swallow. So it's like, I think I just swallowed, but I can't really tell. Yeah, exactly. No, it's fine. You should totally do. Well, okay. So what's cool is then you get to see your airway on the camera, right? You can see it sort of winding down. It's like this branching structure. It feels like you're flying through the Death Star. So you're like flying in and out of it. But anyway, so we got a bunch of healthy smoker, non-smoker volunteers. We took a brushing of their airways, and we took the cells. We used a process called flow cytometry to sort individual cells into wells of a plate that had 96 wells in it. And our grad student painstakingly manually performed this single-cell RNA sequencing procedure. Again, totally glossing over the technical details. Where we were able to get counts of thousands of genes per cell, get a matrix that looks like this. For this particular data set, so we had 6 smokers and 6 non-smokers. We confirmed that they were sort of active smoke exposures based on this test called a Nicoler, which measures cotinine in your blood, which is a metabolite from cigarette smoke exposure. So we have 6, we're really sure that they were actively smoking versus the other ones that weren't. This is, for this particular data set, given that this was several years ago, we were looking at 796 cells after we did our sort of filtering QC, so it doesn't really seem like a big matrix yet, which is true, but the big data part of it will be more towards the end where technology has really allowed this to scale. This was our first one, so we were very excited about these almost 800 cells. And we detected about 4,500 genes that have at least 2 counts in at least 2 cells. So one of the ways in which we like to sort of view the data, to just kind of visually explore it, is through these data dimensionality reduction techniques called TISNEs or UMAPs. Are you guys familiar with that? Yeah. Yeah, it was not exhaustive. So we looked at a possible, I think, 20,000 genes, but a lot of them were just zeros, so we didn't detect them. That doesn't necessarily mean that they're not really there in the cells. It's just sort of the limit of the technology, the sensitivity of the technology. In terms of what was previously done, those sort of bulk studies we're doing on the microarrays, they were more sensitive for genes, so they actually did detect more genes than this, probably about 10,000. But then that's still like an average across all the cells. So it's like you're detecting more genes, but it's an average of the cells, so it's not quite as good of a signal.
So the list that you showed, it only showed the genes where there wasn't a activation?
Correct, yes. Yeah, sorry, I didn't go through the details there, but they measured, I think it was about 10 to 15,000 genes, and then they performed what's called a differential expression test, like a t-test, using linear modeling to see if the mean of the smokers for a gene is higher or lower, and p-value, and do correction multiple hypothesis testing, and those were the final lists that they ended up with. Right, so it was only showing you the different ones. Many genes are not different between the airways of smokers and non-smokers. But for this visualization, we have these. Have people heard of TISNEs or UMAPs before? No, okay. It's a way to view your data in sort of 2D rather than 4,500D. So each point here represents one of our cells, one of our columns in the matrix. And basically, if 2 cells are similar to each other across all genes in the original matrix, so they have a very small Euclidean distance between them, then the algorithms try to find a point in this space where they're also very close together. So these 2 cells are very similar, but very different than those cells over there. So it's a way to sort of view clusters and whatnot. So here we have all of our almost 800 cells, and on this plot they're colored by whether or not they came from a smoker or non-smoker. So you can see that there are clusters, groups of cells that came from the smoker here and here and there, whereas there's other cells that came from the non-smoker, and these are the cells that came from the smoker. And then we can also take this plot and color it by the expression level of particular genes. So FOXJ1 is this gene that's only expressed, it's known to only be expressed in ciliary cells. So here you can see all these cells light up, have high expression of this gene. So we can kind of infer, hey, those are probably ciliary cells that we sequenced. Other genes, keratin5 is a marker for basal cells, so these are probably our basal cell population. Talked before about these secretory cell markers. These are club cells, and these are goblet cells. What's interesting, if you sort of scan back and forth between the 2 plots, you can kind of already confirm what we previously saw with the bulk data, that most of the MUC5AC high cells, goblet cells, are really enriched in the smoker cells, whereas the SCGP1A1 cells are more enriched in the non-smoker cells. And so what we are actually seeing is kind of a shift in the cell population abundances, not just that all genes are sort of simultaneously up and down regulating, or all cells are simultaneously up and down regulating the genes. Yeah.
So smarter controls the expression of these genes biologically, is like electrical process or suppose the control expression of these genes?
Yeah, that's a great question. So there's a whole biological process in how genes are regulated. There are these proteins called transcription factors, which bind to the DNA around the gene. And then there's another, they sort of activate that region. There's another set of called polymerases, which RNA polymerases, which then come on and sort of scan the DNA. And then they create the transcripts, the additional copies. So you have polymerases creating many, many copies. The transcription factors themselves can be controlled by signals from the environment. So you can have a signal, oh, hey, our cell is being exposed to cigarette smoke, and there's a pathway that then activates a particular transcription factor and turns on the expression of these genes.
So theoretically, you could stimulate that without actually causing it.
You could. The problem is, and people do that, not in this, not in my research. People do try to model that with various differential equations or networks or various things. It's very complex because there's a lot of crosstalk between the signaling pathways, and but yeah, that's absolutely an active area of research. So now we wanted to actually apply our clustering algorithm CELDA to it. One quick technical note about numbers of clusters. So for a model, you have to choose the number of cell clusters that you want, number of, which is K, or number of modules that you want, which is L, but we don't know those things a priori. Classic clustering problems. And so we use this metric called perplexity, which is also used a lot in the topic mining or topic modeling field. Sort of a measure of goodness of fit of the model where lower is better. And you can kind of see as we increase K or L, number of cell populations, or each line is a number of modules. Like if we go from 10 to 20 modules, we see a big drop, but then 20 to 30, we get a much smaller drop, and 30 to 40, we get a much sort of smaller drop. And so it's a little bit of a qualitative assessment, but we kind of try to figure out where the drops, the major drops stop happening. Same thing when we're looking at cells, we look to see where it begins to sort of bottom out. And so for this particular data set, we chose L equals 40 and K equals 14. And that was also, there's no sort of statistically perfect way to determine that. It's also looking at it by eye and looking at the biology of each cluster. And so we now have our same plot I showed you before, except for on the right side, or yeah, your right side, I'm showing you the cluster labels for each individual cell group, cell population. And we can label them based on known markers, what we think they are. There's some immune cells. I already mentioned like basal cells, club cells, ciliary cells, goblet cells. There's a group of cells in the middle that we didn't know anything about. So I'll come back to those in a minute, because those are pretty exciting. So just to give you a concept of what a module looks like, so a group of co-expressed genes. This is module number 5. And this is, again, showing you now a similar heat map where I showed you before, where each column now is a cell, each row is a gene, and red means higher relative expression. So you can kind of see that these genes, for the most part, have the same expression pattern across all the different cells, which is why they get grouped in the module. One of the top ranked genes in this module is FOXJ1, which is our marked ciliary cells. When you look at all the other genes too, they're all sort of ciliary components, so they all make sense. And we can estimate sort of the probability of each module in each cell. Sort of the model does that inherently. And then we can see, instead of coloring this plot by individual genes, we can color it by probabilities. See, this is where this module is turned on or off. And then we have 40 of them. So these sort of different 40 components highlight where they're turned on or off in the different cells. And each component should be different than another component, even though some might be slightly similar. So some are very cell type specific, like L1 and 2 there are very expressed in sort of the immune populations. You have some that are more broadly expressed across the majority of populations. So those are genes that are sort of important for every cell to kind of live. There's a set of essential genes that are on in every cell. I mentioned the sort of probability matrices before. That come directly from the model. So this shows you the probability of each module in each cell. Again, it sort of gives you a nice overview of the data set. Now, instead of looking at all cells, you can see sort of what's turned on in what cell population. So this gives us sort of a nice map or a nice atlas of relationship between the genes and really what each cell population is. We can make heat maps that the actual values themselves and see that they sort of correlate. Just two quick interesting findings before I talk about some of the future directions. And the one thing was that there were 3 clusters of, did I step on that? There we go. Which one did I unplug? All right, here we go. We need some more tape on there, I think. So the second, third, and fourth cluster, they were all ciliary cells, so they all express the same module that has ciliary genes. But the fourth cluster, the blue one there, expressed genes in these other 2 modules exclusively. And all those cells came from the smokers. And it turned out when we looked at the genes in there, those were some of the ones that we'd seen, detoxification genes that we'd seen previously being upregulated in our smokers. And so they're pretty specific to these ciliary cells, or induced very highly in the ciliary cells, even more so than the other cell types. And then we could do what's called fluorescence or immunostaining for particular genes to sort of do validation. So there's this one gene called AKR1B10, and look where it was expressed. So I guess over here, so we have a picture of an airway. It might be a little bit hard to see. The yellow there is stained for ciliary components, so you can see the ciliary cells, and they have the little hairs on top. And then the red is sort of showing you the body of the ciliary cells. And you have, in a never smoker, you see very little of this blue, whereas you see a lot in the smoker, and so this blue is showing you sort of the level of that actual gene. So we can kind of confirm that, yes, that gene is mostly expressed or turned on in response to cigarette smoke exposure. There was this other cell population that we didn't know anything about, which, if you look over there, it's mostly in the smokers. Again, we could go back to our probability map and kind of zoom in on a subset of the modules that somewhat define or make that module distinct from the other ones, or make that cell population distinct from the other ones. And it's an interesting one because it's kind of in between 2 other populations. Here we have our basal cells, right next to it we have our basal cells in the smokers. You can tell that it turns on, expresses a lot of the same genes that are turned on there. And then on the other side we have our goblet cells, which also express many of the same modules. However, when you look at the 2 modules up at the top, L16 there, that's turned on in the goblet cells, but not turned on in our cells or the basal cells. And then you have this module in the middle highlighted in red that's turned on not in the basal cells, but it is turned on in the goblet and our sort of unknown cell type. So it's this weird sort of combination of modules that uniquely define this particular cell population. And there's one gene in the module 18 called CCAMP5, and so we did staining on that, and as well as for the goblet cell marker on 5AC to kind of get a better sense of what was going on. So again, we have sort of our airway from never smokers. Each of these gray things represent a cell, just in general, and then green is our CCAM5 marker, but blue is the MUC5AC marker, so that's our blue or goblet cells. Here you can see a smoker. This is a region of what's called goblet cell hyperplasia, so you see all these blue mucus-producing cells just sort of expanding everywhere. But what's interesting, we find cells that only have the green but not the blue expression, these kind of mysterious cells, we find them sort of enriched in the smokers. We don't actually know, or smoker cell goblet hyperplasia, we don't actually know what the cell does yet, so that's sort of our future work is this cell that's sort of transitioning from a basal cell to a goblet cell? Is it a goblet cell that's sort of dying off? Has it lost all of its mucus? We're trying to figure it out. But we would have never known about it if we hadn't done this kind of sequencing before, because it was max when we averaged all the cells together in our bulk data. So. Sorry? Almost. I mean, I'd say when we did the staining, which is a little bit less sensitive than the RNA-seq, but it gives you the spatial resolution. We did find examples of cells that were in the never smokers, but there was a huge enrichment in goblet cell hyperplasia. So yeah, it's kind of showing you what the bar plot down there is.
Josh, I've heard that secondhand smoke is almost worse than smoking. I just wanted to hear what your thoughts are and if you've ever do some stats on that.
Yeah, it's a good question. We've actually done studies, not single cell yet, but we've done other sort of bulk studies where we're looking at secondhand study. Obvious Group has done a lot of it. So secondhand smoke exposure for former smokers and seeing what they look like. You can definitely detect some of these same genes that turn on in smokers. You can detect them in secondhand smoke exposed people as well, although maybe they're not at quite a high of a level. So, in terms of what the sort of health risks are, we can't necessarily say that based on the gene expression alone. That's more of the epidemiological studies where they do show an association. There's an increased risk for other bad things happening, like lung cancer, COPD. But yeah, exactly. People are really interested in that. We do. We had a study published about e-cigs, and there, again, we can't really declare them safe, but they do seem to have less of an effect than full-blown cigarettes, at least when you look at the gene expression profiles and what's being turned on or off. Whether or not, right, that means that there'll be less of an incidence in things like cancer, something like that. We really don't know yet. Yeah.
So, looking at this, I see current smoker and never smoker. Do you have any data about the duration of how long somebody has been a smoker?
Yeah, that was, we do, so we try to record the best we can, sort of when somebody quits, when their last cigarette was, although it's a little bit, when you're asking somebody when their last cigarette, we get a little bit of variability. Sometimes self-report isn't the most reliable thing. Right, so we do see that some genes kind of turn off very quickly after you see smoke exposure, whereas other ones kind of take a lot longer to turn off, months, and then some even remain persistently altered. Actually, the kind of novel cell population has genes like the CCAM5, these other ones, there's sort of some of these genes that are persistently altered. So we're really interested in what that kind of means in terms of long-term kind of dysregulation of the airway. Really, yeah. There's some interesting names. Some people get creative sometimes. I don't think there's any, like there's Chad, there's Hedgehog, people like Sonic at the time, so there's the Hedgehog pathway. Yeah, they're really weird. Most of the time, sometimes if you're lucky, it's related to the function of the gene. Like they're, like a mucus cell, some of these are mucins, and so they have like MUC1. Okay, that kind of makes sense. 1, 2, 3, 4, 5. Other ones are just, have some weird ID or not named very consistently. That's actually, gene annotation is a major problem in and of itself because sometimes multiple genes have the same name, and where on the DNA sequence the gene starts and ends, there's multiple different databases out there that annotate it different ways. So genes are really more a pain. Since we're getting close to time, I'll just finish up, I guess, with a, we think this might have somewhat of an implication in lung cancer because we're also interested in, it's called a subset of lung cancer, which is called lung squamous cell carcinoma, which is thought to originate or develop in your airway. And there's these what are called pre-malignant lesions, these dysplasias and metaplasias, which are sort of like abnormal regions in your airway that some of them may progress on to lung cancer, although most of them will go away or not do anything on their own. But when we stain for specific kind of markers here, and we can look at it across a spectrum, again, here we have the blue is the our goblet cells. We can kind of see them at our hyperplasias or mild dysplasias, but as we get more severe, we see a loss of these blue cells, but we see this sort of persistent green CCAM5 positive cell, and we think that they may have some sort of indication for lung cancer development. It's another sort of area of active development. So just to summarize this part, cell that can identify transcriptional modules and cell populations, and then when we applied this to our smoking data set, we found that a specific set of genes were upregulated in ciliary cells, and we found this sort of novel indeterminate cell population. I'm not going to go through the other parts just because I think we're getting time, but we're really expanding, and this is just sort of the beginning of the data. There's a lot of, there's ways that we can now measure multiple things on this multiple data. So imagine multiple matrices now for each of your cells. How do you integrate that data and use that to characterize and cluster your cells? So really interesting problem. Usability, so for computational people dealing with computational packages, maybe they can analyze it, but there's a lot of biologists out there who want to use the data, so that's where we're using things. We've developed something called a single cell toolkit, which you guys are intimately involved in now. We're really excited about how to sort of scale that, put our algorithms within. It's basically an R-Shiny app. That way non-computational users can access them and perform analyses. And also scaling, so there are data sets out there with millions of cells, and so a lot more than our 800 or so. I help run the single cell sequencing core on the Med Campus, and there are Microfluidics devices now which can generate 100,000 cells in a single run. So the ability to analyze this computationally and memory efficiently and quickly is becoming a bigger factor. And I just want to say a huge thanks to the SAIL group also for helping us get our R01, which has involved methods for developing how to do this sort of multimodal clustering, but also how to sort of build and optimize the packages. I heard Bob Andre isn't here. I heard that he was floating around a slide with a big picture of my face on it, so I wanted to do that back and put a really big picture of him, but I guess he got out of it. So he helped us, I think, without his expertise and his letter of support. Our second aim, which was to develop these sort of cloud-based approaches and how to really scale these type of analyses, we wouldn't have been credible without their input. So it's been an exciting project, and we're looking forward to working with you guys more on it. So yeah, with that, I'll stop there, I guess. And I'm happy to take any more questions or discuss anything else afterwards. Thanks. I would give a lot of it to Doug Densmore. I don't know if you guys work with Doug a little bit. So he's helping us develop. I don't know if you guys went to the talk last time. Allison Segro had really cool videos, and so I wanted to try to throw one slide in there with some videos. Unfortunately, we don't have time to get to it today, but not all of them are running on. Keep going. Anyways, he's building us a new Microfluidics device. So there are some commercial ones available, but they don't do everything that we want. And, I want we're dealing with clinical samples, which are really small and hard to work with, and so the ability to be able to sort out certain cells or profile a larger number of cells cheaply and cost efficiently. He's sort of working on that with us. So at least give some money to him. Other than that, I would just keep profiling. I have a lot of interesting studies going on. I mean, some of them are already grant funded, profiling different cancer specimens, lung cancer, prostate cancer, coming out of patients from EMC. So we're trying to learn about how does the immune system affect cancer development. There's a lot of therapies out there now that are trying to target and activate the immune system to get them to attack the cancer cells. And so that's an interesting area that I'm trying to pursue. Sorry, can you say it one more time? Yeah. Yeah, it's a great question, and we've been thinking about this very deeply. We don't know the answer to it yet. What we think may be happening is that there's a particular kind of mutation that can happen. So you can have cancers caused by mutations to your genome. So you have your DNA sequence. You alter it at specific spots, and it can activate or deactivate certain genes, and those cause cells to start growing uncontrollably. We think there's a mutation in a gene called Notch1, which may cause that particular phenotype or a particular pattern where this CCAM5 positive, this unknown cell sort of expands. But we think that probably only happens in the setting of cigarette smoke exposure. So we're trying to disentangle what those signals are. Just actually submitted a grant a couple days ago to do that. So, It may be, it's in itself, it's probably not enough to become cancerous, but it may increase the risk of getting additional mutations that will then be enough to allow the cells to develop into cancer.
Do you suspect that you might see that same group of cells in people that have lung cancer who don't smoke? Because granted, that population is exceedingly small.
Yeah, it's probably not actually. We think this particular is something specific to smoke exposure and cigarette smoke exposure. Though non-smokers actually have a distinct set of mutations, and we probably won't see this population of cells in there. We might guess. We haven't done it, so that would be my guess. Cool. All right. Well, thank you, everybody.
Thank you.